Hello friends, I am Abhishek Sharma and I welcome you to our YouTube channel Soma's Evolution Classes. In this video, I will brief about pharmacodynamic aspects of drugs, its importance along with its types and various factors affecting pharmacodynamic profile of drugs. Pharmacology involves the effect of drugs on living systems. Comprehensively, it deals with the interaction of exogenously administered chemical molecules or drugs with the living systems. Pharmacology of drugs can be studied under two domains named as pharmacokinetics and pharmacodynamics. Pharmacodynamics basically is the combination of Greek word pharmakon, which means the drug, and the dynamics, which means action or power. In short, after administration, whatever a drug does to the body can be termed as pharmacodynamics. Broadly, pharmacodynamics involves various aspects related to mechanism of action, site of action, duration of action, and type of effect of drugs. Mechanism of action explains how and where the effect is produced along with the type of effect or action produced by the drug. Drugs can mediate their action by stimulation, depression, irritation, replacement, or by cytotoxic type of actions on cellular or subcellular levels. For an instance, rubefacient action of painkiller sprays or balms is a type of irritation type of drug action, which suppresses the feeling of pain sensation going on into the brain. One more instance for replacement type of drug action is treatment of diabetes mellitus by administering insulin as a drug for its unavailability or diminished quantities in the body of the patient, which means the administered insulin as a drug from outside the body replaces the depleted insulin inside the pathophysiology of diabetes mellitus. Mechanism of action can be classified as receptor mediated or non-receptor mediated on the basis of how a drug produces its pharmacological or therapeutic effects. Receptor can be termed as macromolecule or binding site or platform located on the surface or inside the effector cells that serves to recognize the signal molecule or drug and initiate the response to it, but itself it has no other function. Basic functions or basic components of receptor includes recognition of specific ligand molecule, that is the ligand binding domain available onto the surface of the cell membrane. It is generally present in intercellular regions, that is above to the surface of cell membranes. Transduction of signal into response by stimulating a cascade of biochemical events done by effector domain, which is generally present in the intercellular regions, that is inside the cell. When we talk about receptor mediated drug action, interaction of drug and receptor takes place to form drug receptor complex, which is then responsible for generation of pharmacological or therapeutic effect of drugs. This interaction of drugs with receptor takes place on the basis of few basic properties of the drugs named as selectivity, affinity, and intrinsic activity. Selectivity is defined as the ability of a drug or physiological molecule to bind on a particular kind of receptor, that is its degree of complementary correlation between drug and a particular receptor. Affinity can be defined as the readiness of a drug to get bound to the selective receptors. Intrinsic activity is defined as the ability of a drug after being bound to the selective receptor to initiate the cascade of biochemical changes which finally leads to the generation of pharmacological or therapeutic effects. Based upon these three basic properties of drugs or physiological molecules, we can classify drugs broadly into four types or four categories. Agonists, which means these categories of drugs have same affinity and intrinsic activity for the receptor similar to that of physiological molecule. Antagonists, which means these categories of drugs will serve as receptor blockers, as these kind of drug will be having same affinity similar to agonist but zero intrinsic activity. they readily bind to the particular selective receptor, but they are having no potential to generate or initiate the pharmacological effects, which means the receptor gets blocked for other ligands or drugs. Partial agonists, which means these categories of drugs have similar affinity to that of agonists for receptors, but the ability to produce the effect is half in comparison to that of agonists. These drugs produce submaximal effects in comparison to that of agonists. Inverse agonists, which means they are having similar affinity to that of agonist and they readily bind to the receptor but having opposite intrinsic activity in negative values, which means it will produce an effect which is opposite to that of agonist. Receptor mediated drug action is achieved by four types of receptor families: ligand gated ion channels or ionotropic receptors, G protein coupled or metabotropic receptors, enzyme linked receptors or tyrosine kinase linked receptors, receptors regulating gene expression, transcription factors or steroid receptors. So, in this area we will first go to the ionotropic receptors. Ionotropic receptors are localized on cell membrane and coupled directly to an ion channel. These cell surface receptors, also called ligand gated ion channels, enclose ion selective channels for sodium, potassium, calcium or chlorine within their molecules. Ligand binding opens the channel and causes depolarization or hyperpolarization and changes in cytosolic ionic composition depending on the ionic flow through the pharmacological or therapeutic effect is achieved. Next comes the figure where you can see the ionotropic receptor where the ligand binding domain is on the intercellular region or above the cell membrane and the middle portion of the receptor is embedded into the structure of cell membrane. Effector domain is present intracellularly, that is inside the cell. If we talk about the structure, it consists of 5 subunits comprising of 2 alpha, 1 beta, 1 gamma and 1 delta subunits. Next is G protein coupled receptors that are membrane bound, which are coupled to effector system through GTP binding proteins called as G proteins. Effector domain works through main 3 systems. They can work through adenyl cyclase CAMP system, phospholipase C inositol phosphate system, ion channel systems. So, all these 3 are the system through which they can work. When a ligand binds, the receptor activates the attached G protein by causing the exchange of GTP or the red area, or for GDP the yellow area. The activated G protein then dissociates into an alpha and a beta gamma complex. Both activated G protein and beta gamma complex have ability to produce pharmacological or therapeutic effects. Enzymatic receptor or enzyme linked receptors are made up of large extracellular ligand binding domains. It is connected through a single transmembrane helical peptide chain to an intracellular subunit having enzymatic property. Effector domain is directly linked to tyrosine kinase initiated cascade of events. Examples include insulin receptors, epidermal growth factors, nerve growth factors and many other growth factor receptors work in this fashion. Receptor regulating gene expressions are intracellular or cytoplasmic or nuclear receptors, soluble proteins which respond to lipid soluble chemical messengers that penetrate the cells. The ligand receptors dimers move to the nucleus and bind other co-activator or co-repressor proteins which have a modulatory influence on its capacity to alter the gene function. All steroidal hormones, glucocorticoids, mineralocorticoids, androgens, estrogens, progesterone along with thyroxine, vitamin D, vitamin A function in this similar manner. Apart from receptor mediated drug actions, some of the drugs may act through non-receptor mediated chemical or physical actions via other modes. For an instance, antacids performs basic neutralization reaction by neutralizing the acid present in the gut with the help of base present in its composition. In such cases, no receptors are involved in pharmacological or therapeutic effect of the drugs. There are various factors which can modify drug action either quantitatively, that means plasma concentration or the drug action is increased or decreased, or by the qualitative, which means type of response is altered, example drug allergy or idiosyncrasy. Body weight, age, gender, diet, routes of administration, presence of any other disease, tolerance, tachyphylaxis, drug interactions and genetic makeup are various factors which affects or modify the drug actions. That was all about the basics of pharmacodynamics. If you enjoyed this video, please do like and subscribe our channel Soma's Evolution Classes, and for more interesting videos, stay tuned with our channel. Thank you.